From Microbe TV, this is Immune, episode number 83, recorded on August 19th, 2024. I'm Vincent Racaniello, and you're listening to the podcast about the body's defenders against disease. Joining me today from Ithaca, New York, Cindy Leifer.
Hi, and welcome back.
How's it going up there?
It is getting
far north.
Well, far north. It's raining today. It's not that far north. But the students are arriving back, so it's hustle and bustle right now.
Also joining us from Madison, New Jersey, Brianne Barker.
Hi, great to see you all. Our students don't return until the end of this week. Classes start next week, but we will certainly be getting hustle and bustle as well. It's not raining here, but it has been a lot, and it's exciting that the power is currently on because that hasn't been a guarantee.
Yeah, we missed a TWIV a week ago because of no power and TWIP at the same time. Oh. Yeah, you're at the vagaries of technology. It's life. You know, we depend on it, but it's fallible.
Yes. Well, it turns out that at AAI, one of the vendors was giving away flashlights. And that was actually the only flashlight I could find last night. So I was very pleased that I had a flow cytometry flashlight.
Well, about, 2 years ago, someone from New Zealand sent me at the incubator a Bluetti backup battery. Right?
Nice. Yeah.
Oh, wow.
It was like 50 pounds. It's huge. And I brought it home because the power is stable at the incubator. So last Friday, a week ago, the power goes out, and I pull this thing out, and it only has 220 volts AC out.
Oh, no.
I didn't have you need a converter, right?
Right.
Yeah.
So that wasn't, it was fully charged, it was ready to go, and wait, this plug doesn't fit.
Oh, no.
So I don't know what I'm going to do with it because I don't want to have a converter, and that makes noise, blah, blah, blah.
Right.
Yeah.
I'm not even sure I could run everything because you got not only the setup here, but the internet has to be powered, right?
Yeah.
Okay, today we have snippet and paper for you, all courtesy of Cindy. But before we start, let me remind you, if you enjoy these programs where we talk about science and power outages and other things, it's life, right?
Yeah.
We'd love your support. Microbe TV is the production company that does all these podcasts. It's a 501c3 nonprofit, which means at least in the U.S., your contributions are federal tax deductible. Microbe.tv/contribute. Okay, Cindy, take it away.
All right. So I picked both the snippet and the paper today. The snippet I picked because I was at a conference about a week and a half ago that was mostly a not science. It was science adjacent conference, but there were a few scientific talks. And one of them was the senior author of this first paper. And I thought it was just fascinating and not something that we talk about regularly. So the title of the paper is Type 1 Interferon Responsive Microglia Shape Cortical Development and Behavior. So we don't talk about neurobiology much on here, but it's interesting. It's published this spring, April 2024, in Cell. And the first author is Caroline Escobas, and the senior author is Anna Malofsky. And the reason why I thought this paper, so they're at UCSF. And the reason why I thought this paper was interesting is because it is really about a role for type 1 interferon in the development, pretty much, and in the productive repair in the brain following various different insults. And when we think about type 1 interferons, we really think about antiviral responses. And so it was really interesting to think about this in the context of behavior and development in the brain, but yet also a cytokine that we know is really important for antiviral immune responses. So the first thing that we need to talk about is what are microglia, because we've mentioned them, I think, but not in great detail. And I won't go into like all the vagaries of them, but microglia are basically the dominant immune cell type in the brain. And they're located in the parenchyma. And they have a lot of different functions, including, immune functions related to antiviral immunity, but, in production of interferons and things like that. But there are also macrophages in the brain. So they do things that macrophages do, like eat things. And these include pruning and regulating synapses that form in the brain between the neurons to allow for neuronal signaling. And they also regulate neuronal activity. And interesting related to this is they can just basically eat entire neurons, which I thought was pretty wild. And microglia dysfunction is associated with a number of different diseases, including autism, epilepsy, and schizophrenia. And what's interesting is that there is an interferon signature that's associated with microglia in these various different diseases, as well as with aging and other neurodegeneration and neuropathologies.
Yeah. So microglia, it's sort of interesting to me because I think in sort of very basic immunologist terms, immunologists are like, oh yeah, microglia, they're macrophages. Yep. But they're also not totally regular macrophages.
Nope.
They're developing in the brain.
Yep.
And so there's sort of this unique cell type that I think both the immunologists and the neurobiologists take claim to. And so they're not just macrophages, if that's how you think of them, but they do have some similarities to macrophages.
Where does the name come from? Microglia. Does anyone know?
So glia are the cells of the brain that are not neurons.
Okay.
And these were the smaller of them instead of, say, astrocytes.
So there are a lot of them in the brain, right?
So there are.
Okay.
And they're not doing a whole lot in non-damaged brain and non-inflamed brain, but they become activated and they proliferate in the brain as well.
Are we sure that they don't have a huge role in the sort of non-injured brain? I guess I've had this idea that they might be involved more in pruning or clearing up excess neurotransmitter or clearing up debris to keep brain function going well.
So I guess my limitation is I don't know how often that happens. I guess maybe it's happening all the time. So then maybe they are active all the time. But if it's not happening all the time, then they're involved in that when it's happening. So
got it.
But that's my limit.
My impression was that was happening kind of all the time, and they're keeping everybody clean.
Okay, so they're the cleaners. All right. Okay. So they are active all the time, but they're not inflammatory all the time. Let's put it that way.
Yeah.
And that's pretty important because you don't want a lot of inflammation in the brain. Okay. So the other player that's involved in this paper is type 1 interferon. And I always like to make the distinction that this is not interferon gamma. So a lot of people know interferon gamma, and interferon gamma is important for macrophages, but this is type 1 interferon. And so there are two types. There's beta and alpha, and they signal through the interferon alpha receptor to JAK-STAT pathway to induce gene regulation. And again, it's really mostly associated with antiviral immunity. And type 1 interferon is induced by a number of different innate immune sensors, which is why I was interested in this paper, and probably you as well.
Yep.
There's three main types of sensors that can induce it. There's the RNA sensors like RIG-I and MDA5. There's the DNA sensors, one of which is cGAS that triggers sting. And then the third class is toll-like receptors, like RNA receptor for TLR3, DNA receptor TLR9. And the other really interesting thing with type 1 interferons is almost all cells can make type 1 interferon, and almost all cells can respond to type 1 interferon. So there's this way to signal the presence of viral infections through this cytokine through most cell types, including microglia. And so microglia with an interferon signature are actually rare in non-disease states. So this was my idea of they're not doing much in the not inflamed state. But when there is any kind of inflammatory insult, they will upregulate this process. So this group identified interferon responsive microglia, which they called IRMs. And these increase with damage, and they're also present in the developing cortex. So they suggest they might actually have a role in cortical development as well. But they were looking at a damage model, and I'm not going to go into great detail of all the paper, but basically what they do is they take day 2 old pups and put them on ice to induce hypothermia, and they excise a whisker pad. So they go in where they have the little whiskers, and they actually take the whole pad out and the whisker, and then they put on lidocaine, and then they let them sit. And so this gets rid of about 40% of the whiskers, and the neurobiology people will be totally fascinated about this. And I don't know that I fully understand it or understood the piece of data that they showed, but there's topographical rearrangement of all the synapses that lead to those whiskers. So there's basically a remapping of all of the whisker sensation in the mouse when they do this damage. But it leads to increases of neurons with damage, but not synaptic loss. So there's not a major, complete screw up of the brain sensing, but there's a reduction in the arrangement of them. And then what they did was they did single cell sequencing because they wanted to know what was going on a couple of days later after they did this excision. And I thought it was pretty cool when you're doing single cell sequencing, you wonder how many cells can people get, and how are they going to get enough cells to look at this, to look at their the brain of these mice. They have to use 10 mice, and they try to isolate out the microglial cells. They got 12,330 cells that they could get single cell sequencing from, which really, for the original single cell sequencing papers is a lot, but for more recent ones, that's not very many cells.
Yeah.
But
and I will also tell anyone that images here are quite nice. I also don't have a great understanding of some of the 3D parts, but I do understand the one where they show the whiskers and the whiskers being removed. That one I get.
Yeah, that's very clear where you can see where the whiskers are missing, but how the topological rearrangement of the synapses, that was a little above my pay grade. But But the really important thing is they did this single cell sequencing and they found clusters. Everybody finds clusters. And one of the clusters was new. And it was enriched in an interferon signature. So they call these the interferon microglia, the IRMs, interferon responsive microglia. And what's interesting about this is that they could see it conserved. So they found a signature for this cluster. They found it across multiple different diseases, including Alzheimer's, glioblastoma, demyelinating injury, aging, even SARS-CoV-2. So they then went and used a K18 ACE2 transgenic mouse, and they saw that when they did infection, there was an increase in this interferon signature in staining of IFIT-M3 in and around the neurons that were infected, and the microglia lit up. So there is this idea that these are interferon signature expressing microglia. So they're responding to the interferon that's being produced during these different disease states. So they didn't just do SARS-CoV-2, but they also did an Alzheimer's disease model. Mm-hmm. And what they found was that there was engulfment of neurons. So they could see the engulfment of the neurons inside of the microglia. And I thought what was really cool is they use something called RNA velocity trajectory. So it can track development of cells over time based on some splicing variants and things that arise over time. But basically what they found was that there was a cluster that moved toward one that contained neuron mRNAs, suggesting that these interferon responsive microglia were then taking up neurons and moving towards this fate where they've engulfed a neuron.
I'm impressed by your explanation of that, because when I saw RNA velocity analysis, I was a little worried.
I understand it a little teeny tiny bit because we've played around with it, but yeah, it has to do with accrual or buildup of splice variants, and you can line up the sequences over time. You're taking a snapshot in time, but you can kind of get an idea of what cells are at what state based on their accumulation of these these splice variants. Anyway, but yeah, so it seemed to be that these IRMs were then moving towards engulfing these neurons. And interferon was required for this. And basically the bottom line is there are some sensors, including MAVs, which is part of the
RNA sensing.
The RIG-I and MDA5 RNA sensing pathway. They could knock that out and things were fine. CGAS... knocking out of MAVs, but not CGAS or TLR3, phenocopied this idea that the neurons got phagocytosed, but if you deleted interferon, they were no longer phagocytosed. And so they mapped it back basically to saying that it was sensing RNA that was then driving production of interferon that then was causing these microglia to have this interferon signature and then eat the neurons. And apparently part of this that didn't make a lot of sense to me is neurons are high in mRNAs, higher than most other cell types. So I guess engulfment of them also then triggers more RNA sensors that causes more interferon production, and there's a feed-forward loop. But they basically said that interferon production was necessary and sufficient to have this phagocytic process because they also used zebrafish, and they could show if they injected interferon intracerebroventricularly, they could observe the induction of these IRMs, and it reduced the number of neurons in the fish. And I just want to note we've talked about zebrafish immunology on episode 41. So if you're interested in more in zebrafish immunology, it was more, I think we were talking about neutrophils or macrophages then. But it's just interesting. And then the last thing they did in this paper, just quickly, is that they looked at tactile hypersensitivity. So if you touch a mouse's whiskers, they don't like it. They move away because they sense something really close to them. And basically what they showed was that if you knocked out interferon alpha receptor, that it basically increased their tactile sensitivity. So they became more sensitive to the touching of their whiskers. And it has something to do with eliminating neurons that led to increased signaling in the other neurons that were left. So basically what this is saying is that there's innate sensing that increases type 1 interferon in the brain. That type 1 interferon then signals on microglial cells. Those microglial cells can then phagocytose neurons. And they argue that this is important for development in the brain, but also important for eliminating damaged neurons that might happen during inflammatory disease. And they make an important distinction between this kind of eating neurons and efferocytosis, which is when cells die and get eaten, because these are not dead neurons, they're just damaged neurons.
I thought apoptosis was a major way of getting rid of damaged neurons too.
Probably. That's more than I am aware of, but you probably talked about it more. But I think they can be damaged first and then go through apoptosis once they sense their own damage. So this might even be earlier than that. But I don't know how whether the microglia are able to detect that the neuron is damaged or they're just eating them randomly. It wasn't really clear to me.
That also was not clear to me, but I could imagine that you might have a neuron that is damaged, but still kind of trying to signal and signaling aberrantly. And so you want to eat that neuron before it messes up sort of the whole network when it hasn't necessarily gone to apoptosis.
Yeah, that makes sense to me.
I sort of wonder, I think that this reminds me of, ideas of type 1 interferon and other ways that it may impact the brain. And sort of, makes me think about, how many other ways are we seeing interactions between some of these things that we think about a lot as immunologists and some of these other parts of physiology that we're not normally thinking about. I cannot say that I have a great understanding of cortical development in the brain.
Nope.
And it's not something I think about, but I do think a lot about interferon. And so it's interesting to see, this molecule that I'm spending a lot of time measuring in my lab is impacting these processes we don't normally think about. And I think that's cool.
Is it surprising that interferon would have a non-infectious role?
I don't think so.
No.
I think that there are lots of situations where interferon's made during tissue damage that we've been thinking about. And we know that pretty much every cell has an interferon receptor. So I guess it's not completely surprising, but it does, again, kind of make me think about unexpected or unknown or as yet unexplored, perhaps, sequelae of some of these infectious diseases or things that we're not realizing could happen and things that we could intervene on.
Sure. When there's an overlap, it's bound to happen, right? It could be, with SARS-CoV-2, you get so many other sequelae, many of them probably due to inflammation, right?
Right.
Yeah. Well, I was wondering about that because, we think about the post-viral sequelae and think about, it caused inflammation in the brain and that's what caused the problem. But might it be something related to this where if you trigger the production of interferon, either systemically that might get into the brain, I don't know how frequently that happens, but I would think it would, or locally producing it, like if some of the virus gets in the brain, which we know happens now, maybe there's production of interferon that activates these microglia and then they're eating neurons, and that has impacts on various different processes that happen.
Yeah, I could also imagine that the timing could be really important. So if you were to think about, interferon being produced for a short burst of time, then that's one thing. But interferon being produced chronically really could lead to some serious damage.
Yeah. But I thought it was interesting to think about this different role for the interferon, but also then, related to viral infections and what's going on in the brain and these post-viral sequelae. I thought that this paper was pretty interesting in that aspect.
Absolutely.
Something new for me. Yep. Absolutely. Okay. Want to move on to our paper, Cindy?
Sure. So let me get my other notes up. I just closed it by accident. Sorry. Had everything ready to go.
So we were just mentioning apoptosis and we were mentioning efferocytosis, and now we get a new type of cytosis.
Exactly.
Nexocytosis. Which was a new word for me.
What? Yeah, well, it's a new word for everyone because they created it.
Right.
So the name of this paper is Neutrophil Trapping and Nexocytosis, Mast Cell Mediated Processes for Inflammatory Signal Relay. So this one was also in Cell. It is September 19th, 2024, and today is August 19th, 2024. So this is obviously published online before it's published.
So Cindy is a time traveler.
Nah, not yet. Well, we're still working on it. The first author is Michael Mihlan. I'm not sure how you say that. And the last author is Tim Lammerman, and they are at the Max Planck Institute in Freiburg, Germany. This paper made the rounds on X and other socials because it's very interesting. And if you take nothing else out of this little thing, basically mast cells can eat neutrophils and then use them for their own benefit.
So this is a theme today, cells eating cells. Huh?
Yeah. So it is cells eating cells, yes. But this is a completely different way of eating cells. And actually, since you said that, I'm going to jump to one of the things that I wrote later on. So you're right. The last one we were talking about microglia eating neurons, and this is mast cells eating neutrophils. Basically, cell-in-cell structures. I didn't really think about this happening. What's different about a cell-in-cell structure from what we just talked about is the microglia are eating the neurons to get rid of them. And that's one outcome of potential cell-in-cell structures. But I wasn't really aware that there are other ways to do this, and it has a name. It's called entosis. And one of the major things that's known to work this way is a tumor cell can eat a neighboring tumor cell when nutrients get limiting, basically to grab its nutrients and use them.
Cannibalism.
It's cannibalism, right, exactly. So it's cell cannibalism. And this can be homotypic or heterotypic. So you can eat your same clonal cells, or you can eat other kinds of cells. So in this case, we're talking about heterotypic because mast cells are eating neutrophils.
Wow, if we could turn that on in a tumor, that would be cool. Have it digest itself.
That's actually a really interesting.
That's really cool. See, whereas I'm looking at this, and I have been having a lot of allergy symptoms, and I'm mad at my mast cells, and now I want to make sure my mast cells don't get these extra nutrients.
I don't know if I can help you with that one. But we are going to talk about allergies in a minute. But I knew about one type of this entosis that I had never talked about it that way, but I don't know if you know about this, where you know how neutrophils go through blood vessels and they come out the other side, and there's a whole process there. But some of them will dive in through the middle of a cell. So they will dive into a cell and then they will come out the other side of the cell, which I thought was really cool. And when I teach it, the students love that. They're like, oh, wait, it can just go through another cell? Like, yeah, that can happen.
Is there a name for that?
Transcytosis, but it's just not at a junction between two cells. It just goes
it's like from the blood vessel. You mean it's going through? Oh, I see. Okay.
Yeah.
I always thought it went through the gaps, but so it can go right through the cell.
Sometimes they can go right through the middle. Yeah.
Like, I don't have time to mess with these gaps.
I don't have time to look for a hole. I'm just going to go straight through the wall. Yeah. So that's one way of doing it. But so there's a whole bunch of different ways that cell structures can outcomes of that. So one is just that they engulf, and the cell that eats the other cell protects the cell. So examples of this are an astrocyte can kind of like eat and protect an oligodendrocyte. This has been found in MS. There can be the cell that engulfs it can nurture the cell inside. And this happens in the thymus with thymocytes. So apparently there are thymic nurse cells that eat and hold the thymus cells inside of them for a little while and then spit them out later when they mature. It's interesting. You can destroy the host cell, so you can eat and destroy the cell that's inside of it that it eats up. So that's just the norm way of thinking, like those tumor cells that are going to eat their neighbor and chew them up and use the nutrients. But the other way can happen too, which I thought was really wild. So the cell that gets eaten can destroy the host cell, almost like a parasite invading and then feeding off the host cell and then getting back out. So there's this, it's just, I went down a rabbit hole learning about all these different ways that cells can live inside of other cells. And we're going to learn that when these mast cells eat these neutrophils, the neutrophils can survive for a little while. They can even move inside the mast cell, which was just really, really cool.
So it feels like in some ways this is like thinking about, well, you know, many of these things are things that, say, prokaryotic cells can do. And we did at one point evolve from prokaryotic cells. So I guess it's not a shock that our cells kept those abilities.
Exactly.
But to be honest with you, I just still get sort of mind blown by the physics of it all. Of like, how is one cell going to be big enough to engulf another eukaryotic cell?
I know. Well, macrophages do it because they mobilize their internal membranes and they put them up on the surface, and they can eat things bigger than themselves, or they can even eat like many, 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 many things and like fill up and become engorged. But yeah, I mean, something like a mast cell is quite a large cell as well, like a macrophage. Right. But it does come into question, like how big of something can you eat? I don't know.
Exactly.
Reminds me of animals who dislocate their jaws to eat big things.
Yes.
It's like the macrophage with the internal membrane.
It takes those membranes and makes itself bigger. Yeah.
Exactly.
That's cool.
Yeah, it's really neat. All right. So the key players in this are neutrophils and mast cells and allergy responses. So what are neutrophils? We have talked about them a little bit in episode 73. It was called Big Macs. Remember that paper where we talked about the number and mass of all the different cells in the body? If you haven't seen that one, you should check it out. It's very, very cool. But so neutrophils are a highly abundant immune cell type in the blood, making up 40 to 60% of the immune cells. They're produced in the bone marrow, and I thought this was really cool. There's about 100 billion neutrophils produced daily. So there's a lot being made all the time.
Whenever you think you're not doing something, you are.
You're making neutrophils. They circulate in the blood, and as we just mentioned, they exit the blood when there's an infection, and they move around in the tissue through chemotaxis. They know where to go because there's markers put up on the blood vessel, and then they exit the blood vessel right where those markers are. We can talk about that details later if you want. But one cool thing about neutrophils that they also do is they do something called swarming. And this has been described for a number of years, and there's an extensive literature on this. And we only discovered this because people can take tissues and do 2-photon imaging and use mice that have fluorescently labeled cells. So you can have mice that have, for example, neutrophils that are expressing a green fluorescent protein. Then when you excite light on them, all of the neutrophils light up green. And then you can track them in live tissue. And you can watch them move around. And in a parasitic infection, you can see the neutrophils will swarm. Basically, they come from all over the tissue, and they swarm in a big group right around where whatever the damage is. So they're known to do this. And I thought this was cool. There's pioneer neutrophils that are the first ones to arrive, and then they recruit the second wave of neutrophils. And then there's a whole bunch of other waves that happen. And this has been really well studied during infectious responses and tissue damage responses, but not very much known in the context of allergic responses.
Right. So if people have, had a complete blood count or had, something like that done, or they've heard that they have an increased number of white cells in the blood, that's usually because of the neutrophils mobilizing into the blood, just like Cindy mentioned, to deal with an infection. And so usually I think about neutrophils and infection. I kind of think of them a little bit as Pac-Man. Although they do have this additional function where they can make extracellular traps, where they throw DNA and some other molecules out and kind of make a little web to trap things like Spider-Man would do. And so I think that the extracellular traps, so they're making this trap and throwing it outside of a cell, is going to be something to contrast with a different part of this paper. But sort of that role in, particularly in my mind, sort of bacterial infection is what I think of with neutrophils. The idea that I would think of neutrophils and allergy is not necessarily something that I would often think about.
Right. I would not think about that either. We usually consider neutrophils experts at killing bacteria.
Right.
And like you said, they can throw these nets out. They can also eat the bacteria and degrade them inside. Or they can also take they're granulocytes, so they contain granules. They can spit those granules out, maybe not expelling their DNA, but just the granules. And that will also help kill bacteria. Like if they're in biofilms or something they can't eat.
Right.
So there are those different ways of doing this. But we think that's bacterial. What does this have to do with allergy? But if you think about allergic responses, I know I have pretty bad responses to when I get bit by a mosquito or something, right? So I have an allergic response to the mosquito saliva, and then I scratch it, right? Because it itches a lot, and then you damage your tissue, and then you can get bacteria in there, right? So then you're going to have neutrophils called, but then you might have additional allergic responses in the same or nearby place. So there's going to be neutrophils in the tissue already, and then you might have an allergic response on top of that. So I think what prompted this group to look at what they're looking at is the fact that there might be situations where you have neutrophils called in because there was damage that you're repairing or whatever. And then you have this chronic repeated allergic response. And say, what happens to those neutrophils in the tissue? Do they do anything, or do they just sit there, and allergic responses and the neutrophil bacterial responses are completely independent and unrelated? Well, it turns out they're not. And basically, we've talked about allergies before too, and we talked about them in the context of peanut allergy on Immune 70 with the allergy of picky eating. But we're going to talk about, these are type 1 hypersensitivity reactions. So the allergy to peanut is also a type 1 hypersensitivity reaction. And these reactions are happening because you have an exposure to whatever it is that you're going to generate the allergy to. And your body makes antibodies that are of the isotype called IgE. And those IgE antibodies bind to a receptor that's on the surface of the mast cells that's specifically designed to bind the IgE antibodies with high affinity. So we call this coating the mast cells with IgE antibody. And then they just hold on to that and wait. And call this a sensitization because if you never got exposed to that allergen again, nothing would happen. You would just exist perfectly happily forever. But as Brianne knows, if you are, for example, allergic to ragweed and your mast cells are coated with antibodies to ragweed, when you breathe that ragweed again, now something happens. So what happens? Well, the antibody that's bound to the Fc receptor then binds to the antigen. And that is like flytrap paper. It just kind of sticks the antigen and causes the surface receptors to bind multiple of these. And then that signals to the mast cell, you have now encountered the thing that we need to react to, which of course you probably shouldn't have reacted to it unless it was a parasite, and then you should do this. But if it's just ragweed, we should have ignored it, but some people don't. So now the mast cell goes crazy and starts degranulating because it's also a granulocyte, and it starts spitting out the granules, and there's something in the granules that causes most of the problem, and that's called histamine. So that's why we take antihistamines, because the granules of the mast cells are spitting out histamine, and that has a number of different responses on the body that include generating all those things that we associate with allergies, like itching and swelling and redness, vasodilation, smooth muscle contraction. So some people, if they're having an anaphylactic reaction, their throat will close, their lungs will bronchial constrict, and they'll have trouble breathing. Blood vessels dilate, so you can have basically a drop in blood pressure. You can have an increased heart rate. Intestinal issues, all of these things are mediated by histamines that are secreted by mast cells, and mast cells are poised all over the body in your tissues. So all of your barrier surfaces, your lungs and your gut all have mast cells, all of your tissues have mast cells. So the mast cells can be responding when they are exposed to these antigens through the IgE on their surface.
Yeah, and histamine can be sort of potentially useful because histamine being secreted towards, say, extracellular pathogen can help to damage that pathogen. And I am clearly going to have to change how I discuss this with my students, because I usually talk about, it's too big to eat. So you have to secrete things at it to kill that particularly that parasite. Now I guess I don't know what too big to eat means anymore. So, Well, but so the idea is that you're secreting something that's going to damage that eukaryotic parasite, but perhaps it will also lead to some tissue damage. But many of the other things that Cindy was mentioning, I was thinking to myself, maybe this is because I've been working on syllabus a lot lately. Like all of those are things that I think of as classic inflammation, and I always put neutrophils in there. So why did I not put neutrophils with this before?
Exactly. Yep.
So you're saying basically that this IgE mast cell system exists for parasites, but then it also engages allergens, and that's bad. There's nothing good that comes out of that, right?
Correct.
Yeah. I don't think there's any good of having an allergy.
No, I cannot tell you anything that's good about having an allergy at all. I think that there are a couple of different ideas about some of the details of this, but part of it may have to do with cross-reactivity between some of those allergens and some antigens that you may find on the parasite. There's also some question about whether there's cross-reactivity to things like venoms. Or other kind of toxins. All of those things that you want to expel right away, which is what my body tries to do with ragweed.
But not everybody has allergies,
That's right?
That's
right.
But they probably respond to parasites nonetheless, which is the way it should be, right?
Right.
Yeah.
Yes.
Okay.
So it's just that some people are responding to things that are not a problem.
Yeah.
inappropriately.
Right. Yeah. So there is occasionally, and we don't understand why, but there's probably some genetic component to it, some environmental exposure and timing, everything that people are generating immune responses, specifically IgE, to things that should be innocuous and you should ignore.
For sure, peanuts should be innocuous, right?
Yeah.
Mm-hmm. Peanuts should be innocuous. Grass should be innocuous.
Right.
Ragweed should be innocuous. I can make a list of things that.
Cottonwood trees.
Yeah.
They're a big one. Yeah. This group decided, though, that they must have been smarter than we were and thought about mast cells and inflammation, or they just had a creative idea and started looking at this. But they were asking, are there potentially interactions between neutrophils, especially if there is multiple rounds of exposure to allergens and induced inflammation, recruitment of neutrophils, and then mast cells are then, spitting out their histamine and degranulating when there are neutrophils around. Does anything happen? So they use this really cool model where they can do in vivo imaging, and they labeled mast cells with one color and neutrophils with a different color. So mast cells they labeled with TD tomato, which is a red, and neutrophils they labeled with GFP, which is green. And they do two photon intravital imaging to look at these cells moving in live mouse ears, because ears are pretty thin and you can just put a objective down on the ear and you can image the cells moving around. And there's not much happening. Neutrophils move around the tissue, mast cells stay put. But if they sensitize the animal to have an allergic reaction, then things get interesting. So the way they induce the allergen is they inject locally antibodies that are IgE isotype that are to something called DNP-BSA. So it's dinitrophenyl bovine serum albumin. It's just an antigen for these purposes. But they have IgE specific for this DNP-BSA. They put them in the ear. Now all of the mast cells are coated with this anti-DNP-BSA IgE, and they're sitting around, still nothing going on, pretty boring. But if they inject IV this antigen, DNP-BSA, now all of a sudden those mast cells in the tissue in the ear will degranulate. And it totally changed the migration of the neutrophils. They swarmed. They swarmed in, I guess, like Taylor Swift fans, Taylor Swift, right around the degranulating mast cell. And they form these clusters. And it happens really fast, within 10 minutes. And they hang around for a while and they eventually dissolve. Meaning the cells don't dissolve, but the clusters dissolve and the neutrophils move away, which would be fine. This also happens with different insults. It's not just allergic responses. They did ischemia reperfusion. It also caused the neutrophils to swarm. And they were also able to show that the neutrophils swarmed if they embedded neutrophils and mast cells in collagen gels and then induced the mast cell degranulation. They could do this too. What's really fascinating, and why I thought this paper was super interesting, as many other people did, is that not all the neutrophils left and went back about their business. Some seem to get stuck right near the mast cells. And when they zoomed in and they did confocal microscopy and transmission electron microscopy, they found that actually the neutrophils got trapped inside the mast cell. So the mast cells ate the neutrophil. And you'd be like, okay, so maybe there was a dead neutrophil there and the mast cell ate it up. No. When they did live cell imaging, they actually could see the neutrophil still moving around inside the mast cell. So these are, these cell-in-cell structures that we started out talking about. So there's something unique about this, that the neutrophil actually lives inside the mast cell, at least for a short period of time. And they call these now mast cell intracellular traps, or MITs, as opposed to neutrophil extracellular traps, where there's this Spider-Man shooting out the DNA to trap bacteria by neutrophils. These are actually mast cells now trapping the neutrophils inside of them.
So is it since neutrophils can cross cells, is it the neutrophil going in or the mast cell grabbing them?
So I think it's a combination of both, because they do go and look at how this all works, like how do they get inside? First, the question is, does it really happen? Like, is this real? right? Because I had some concerns. You do some microscopy and you say something's inside of something else. It could be wrapped around, right?
Yeah.
You need to make sure that it's really inside. I'll tell you, they pretty much convinced me that that was real. They do actually, they do transmission electron microscopy. They see these things are stable up to like 48 hours. So it's definitely getting inside. The other important thing that we need to mention is that it does happen in human. Because, so what if it happens in a mouse ear, right? They can't say how frequently it is, but they did see these MITs, these mast cell intracellular traps in tissue from patients with spontaneous urticaria and Schnitzler's syndrome. So it definitely happens, and they could induce it in human cells. So that's all happening. But, coming back to how do they get in there is the mast cell eating the neutrophil, or is the neutrophil diving in the mast cell? To get at this question, they asked whether the mast cell needed to degranulate, because the model was they coat the mast cells with IgE and then they trigger them to degranulate, and then they see these neutrophils swarm and they get some of them trapped inside. So the question is, do they need to degranulate?
Right. And to me, my idea before all of this would have been to say, oh, well, it must be that the neutrophil is trying to get in. And the reason why I would think that is because I don't usually think of mast cells as being phagocytic. And so I was like, well, it definitely isn't that the mast cell is internalizing. It has to be the neutrophil, because we've heard of the neutrophil going into cells before, and mast cells are definitely not phagocytic.
Yeah.
Would be kind of my thought if I were them trying to answer that question.
Yeah, well, some of a little spoiler alert is one thing I didn't realize is that when these mast cells degranulate, they can have what's called compound degranulation, where a whole bunch of the granules go to a similar location in the membrane and start to degranulate. And what it does is forms a channel inside of the mast cell. So part of it is that the neutrophil is going in that channel. It still comes down to, are they going in willingly, or are they purposefully going in, or are they being dragged in? I don't think that this paper could truly address which one of those was happening. I think we need to know a little bit more about the mechanism before one could figure that out. You'd have to knock something out on the neutrophil, and if that prevented it from going in, then it was the neutrophil. But
yeah, and you'd have to also kind of think about looking at cell biologically, is there a cytoskeletal rearrangement that is similar to what happens with phagocytosis, or is this sort of like a really big endocytosis, and do you see some of those proteins involved or something like that?
So they did a little bit of that.
Right.
So the first thing they asked was, do the mast cells actually need to degranulate, or could they be secreting something else that's responsible for this? To do this, they use a mouse that's deficient in a protein called UNC13D. And basically, the mast cells from these mice are really wimpy degranulators. They just spit out a little bit of their granule contents, but they don't do a full degranulation. And when they did that, they found far fewer MITs. So that would argue that you do need the degranulation to have the MIT formation. But what was interesting is that the neutrophils still swarmed, and they still made contact. They just didn't get eaten. So there's something separating the swarming and the contact from the internalization. And the internalization requires something that's in the granules, which was interesting.
Yes.
So what is it in the granules that's responsible? So they tried an interesting experiment. So they degranulated degranulated the mast cells and then waited before they put the neutrophils in. And if they did that, if they waited 30 to 60 minutes, there were no MITs. So that told them, well, there's something that's secreted very early on following degranulation that was responsible for this. So they said, we already know one thing that does that, and that we already know is important for swarming, and that's LTB4, or leukotriene B4. It's a lipid mediator made by also neutrophils, which is going to come back and be important in a minute, but also mast cells. And it's already been shown that LTB4 is important for neutrophil swarming. So that was a good guess. And if they looked, mast cells will make leukotriene B4 within 4 minutes. They could detect it. So super fast. Whereas some of things like cytokines and chemokines are secreted more like detectable as early as 20 minutes later or a couple hours later. So they said, okay, let's test the role for leukotriene B4. So how do they do that? Well, they could block synthesis of leukotriene B4, or they could block response to leukotriene B4. They could knock out genes that are required for leukotriene B4 synthesis. All of those reduce the number of MITs that were formed. So that told them that there is an important role for leukotriene B4 in this process. Now, it gets a little complicated because neutrophils can also make leukotriene B4. And neutrophils can also induce their own swarming. So if you remember, I told you there were pioneer neutrophils that started a swarm. And guess what they secrete to recruit all their friends to swarm? It's leukotriene B4. So if the neutrophils can also make it, then blocking it or blocking production of it has a little bit, it's a little bit questionable. They
could
tried
be blocking a few different steps.
Correct. You could just be blocking the neutrophils themselves. So they did a few things where they blocked the ability of the mast cells to make leukotriene B4, and they incubated them with wild-type neutrophils, and they still saw reduced MITs. So that suggests probably that it is the mast cell production of leukotriene B4 that's important. And if they incubated wild-type mast cells that can make leukotriene B4, and with neutrophils that can't respond to it, it also reduced MITs. But that one has a little bit of a tricky thing because leukotriene B4 is important for just general swarming of neutrophils. So if you knock that out, it's like kind of a weird thing. So they tried to get around that by recruiting the neutrophils first with a different mechanism and then triggering degranulation and looking. But it gets a little complicated in the weeds. And if you study this, you should go look at it because I still think it's a little bit calls into question their conclusion. But they argue that there's a role, at least a role, for leukotriene B4 in the MIT formation, meaning that the mast cells eating and holding on to the neutrophils. So they provide some mechanism of how this all works, but coming back to like how do they actually get in? That's a different question. What I thought was important that they asked was, is it only neutrophils that mast cells eat, or can they eat other things? And can they eat more than one thing? And which things do they eat?
Hoping that if you weren't going to mention this figure, I was going to make sure we did, because I think this, like when I looked at this figure for the first time and sort of stared at it, it was like, whoa.
It's really, I mean, it's cool. If you have a chance to go look at these pictures, because what they show is that the data are basically that they can eat neutrophils, they can eat eosinophils, they can eat neutrophils and eosinophils at the same time, one of each, but they do not eat monocytes or dendritic cells.
How about basophils?
They did not try basophils.
That's the logo of immune, right?
Yes,
they
did not try basophils. And I have two thoughts about that. One, I think that would be a particularly interesting experiment because there are some questions about the relationship between mast cells and basophils in terms of lineage. And so it would be interesting to know whether that eating happened and how that might, do they eat their progeny or their progenitor or whatever,
Do they eat their young?
Right, yeah. But at the same time, there are so few basophils in an organism that I can imagine that experiment. I mean, just doing the experiment that they've done with, say, eosinophils would be a challenge, but there are so few basophils. I just don't know how you would actually pull that off.
All right. So you could isolate mast cells and you could isolate basophils and you could incubate them together and see if they would work. The question that I have is, when will they encounter each other? Because basophils are really mostly blood cells.
True.
So the eosinophils will, hang out in tissues or go into tissues and stay there. And the mast cells are tissue resident. So yes, I'm sure there's conditions where there's blood vessel breach and there's basophils that get out into tissues, but I think it's not very frequent. but maybe a listener can correct me. I'm not a basophil aficionado. So we know that these mast cells can eat a couple different kinds of cells, but not monocytes or dendritic cells. It's also interesting that you can induce degranulation by mast cells a couple of different ways. So they did using compound 4880 to induce mast cell degranulation. And that induced, here's another interesting thing, a reasonable amount of LTB4, but very few MITs. So my takeaway from the complex experiments they did to try and block LTB4, block signaling to LTB4 and mixing cells together, and then looking at different degranulation and seeing that a different kind of degranulation can also induce LTB4, but not very few MITs. I think the jury's out on the role of LTB4, and it's probably not acting alone.
Yeah, I think that all of this taken together does say there's something about signaling through that Fc epsilon receptor.
Yeah.
There's something about the IgE that's being bound to the mast cell and the signaling that's coming in from that IgE and the Fc receptor. And that could be in combination with the LTB4, that could be separate, but there's something special about the IgE.
Yeah. The next thing they addressed was coming back to what Vincent was asking, is how the heck does the neutrophil get inside the mast cell? And is actin involved? Is it actin cytoskeleton rearrangement like we typically think about phagocytosis? Well, one of the first things they asked was what happens to the mast cell when it degranulates? And it turns out that it gets soft. So it has reduced elasticity of the plasma membrane once it degranulates, which is kind of cool. So it completely changes its physical properties. And they were able to get images of mast cells basically reaching up and wrapping around a neutrophil. So it takes its membrane, it reaches up and it wraps around the neutrophil. And then the neutrophils tend to go in, I mentioned these compound exocytic channels. So when they degranulate, these form these channels, and the neutrophil tends to go into those channels. So there's a combination of the mast cell reaching up and wrapping around its membrane, but also the neutrophil sliding down into these channels. So it's not exactly phagocytosis, but they try to get at this a little bit more. So they use their fancy labeling techniques again. This time they labeled the plasma membrane with TD tomato, and they labeled the actin, LifeAct, GFP with GFP. So they could look at the cytoskeleton and the plasma membrane at the same time. And what they could see is they enumerated two different modes of uptake. So 3/4 of the cells went through the first mode, where there's a sequential membrane tethering. So the mast cell grabs onto the neutrophil. Then it forms an actin cup like it's a normal phagocytosis. But then the actin cup resolves and dissolves, and then there's complete internalization. So they suggest that it's sort of an actin cup that forms, but it's not the typical phagocytosis that you think, where the actin cup comes up and then it forms all the way around and then the plasma membrane pinches off. So it's a little bit different. The second mode, which about 1/4 of the cells go through, was less clear to me. I don't know. They said there's a membrane and actin worked together at the same time to engulf the neutrophil. I think that's a little bit more typical of phagocytosis, but their images weren't as clear to me how that second process worked.
Yeah, it was not very clear to me either.
Okay. Regardless of how they got in, once they were in, they saw co-localization of mast cell granule components with the neutrophils, and they took this to mean that the neutrophils were going into where these compound exocytic processes were, and then being pulled in and being in close proximity or connected directly to the granule compartment of the mast cell. So that's kind of interesting. Well, I would have thought that would mean that there's probably degradative things in those granules and they would chew up whatever's inside, but
right.
They do see them like live for 12 to 48 hours. They can see them swimming around inside.
Mm-hmm.
It's very interesting.
Yeah, no, I don't really know entirely what to make of that. I think that when I was thinking about this section, I was thinking a little bit about this image I use every time I teach about this of a mast cell, and I have it's a electron microscopy image. There's where you can see a mast cell full of granules, and then you can see the mast cell after it's degranulated.
It's amazing.
Right, it's amazing, and
they spit them all out.
They spit them all out, and they look dramatically different.
Yeah,
and I think about that image, and then we think about some of the things that they talk about here, like the mast cell becoming soft.
Mm-hmm.
And I imagine that degranulated mast cell that I show every time, and I'm like, yeah, I buy that cell is pretty soft compared to the other one beforehand. Like, yeah, absolutely. Or, thinking about the sort of size of these channels or the size of these resultant spots. I don't know what we would say, but again, I think back to the image and I'm like, oh yeah, totally.
Yeah.
I totally see those in that image that I show all the time. And so I look at this and I'm like, wow, in some ways I feel like, why didn't we catch that before if we've been looking at these classic images for so long? Because it does make a lot of sense when you actually think about what we've seen for many years.
Yeah. Big bags of marbles that once you spit all the marbles out, it's just a big gooby bag. Yeah.
With sort of a very uneven surface.
And what's going to become interesting next is, so what happens to that mast cell after it does that? I mean, it spits everything. It lays it all out on the table, right?
Mm-hmm.
But they last long term in the tissue. So what happens to them?
I know, unfortunately. If only they would last short term, then I'd feel much better.
They remake all of this stuff. Okay. That takes a lot of energy. So they started to say, I wonder why a mast cell would take up a neutrophil.
And
maybe, just maybe, it's a source of nutrients to remake all of the stuff that it needs to remake.
They also, in the text, the title of this section is excellent because they call this section, mast cells are the winning cells of MIT formation. And I don't think I've ever been able to write about winning cells before, but now I want to.
Yeah. I think it's because they thought about that cell-in-cell endosis that has these multiple different fates. And one of the fates is that the engulfing cell wins.
Yes.
It's probably the most common fate.
Probably, but I just like talking about winning cells.
Yeah. It would have been really interesting if the neutrophil won.
Yeah.
But
Is it reasonable to use the cell as a source of nutrients?
I think so.
Yeah. I mean, I think that it's not unheard of. After neutrophils do phagocytosis of bacteria, they tend to die and get phagocytosed by other cells. And we often think about that as being nutrients.
Okay.
I guess if we think about it, there are a lot of neutrophils. They're short-lived. We're making lots more all the time. So this, I guess, seems like a nice recycling.
So that's, in your opinion, the main function of this.
It's going to get more interesting in just a minute.
Okay.
Because yes, it could be that they're just eating them to use up the nutrients. However, they might also gain new functions, which is kind of cool. The advantage that it provides, the argument that they make, that the advantage it provides to the mast cell is that if they look at proliferation, for example, the MITs proliferated more than the mast cells alone. The MITs had higher viability than the mast cells alone. And then they wanted to look at whether there were any different functions or gene transcription profiles that were different in the MITs versus the mast cells alone. The problem here, and I thought this was like the RNA velocity from the last paper, I think this is one of the coolest things that I had seen because I didn't know that this was a way to solve this problem. So the big thing that everybody does is single cell sequence stuff or sequence things, right? And in this case, it's just some sequencing. But how do you distinguish if one cell ate another cell, whose RNA is whose? Right? That's a problem. So these are a really cool thing. So they use a ribo tag. So this is hemagglutinin tagged. So hemagglutinin from influenza, tagged ribosomes. So it's a transgenic mouse or a knock-in mouse where the ribosomes all have an HA tag. Now you can take wild type neutrophils with ribotagged mast cells, and you can, sensitize the mast cells and cause them to degranulate and eat the neutrophils. And now you can isolate the RNA from the neutrophils that have eaten from the mast cells that have eaten a neutrophil and from the mast cells that have not eaten a neutrophil. And you can look at the translatome because it's the mRNAs that are associated with translating ribosomes. So you can isolate the ribosomes, pull the RNAs off, and do sequencing. So they were able to do this, and they were able to isolate the translatomes of the MITs and compare them to the mast cells themselves. And what they observed was an increase, go imagine this, increase in Fc epsilon R1 signaling pathways. They also saw oxidative phosphorylation pathways and translation signatures. So there's something different about the signaling within the mast cells that have neutrophils versus the mast cells that don't. And since they saw that these various different signaling pathways, they asked, might this endow these mast cells with additional new functions or better functions that they already do? So the first thing they asked is, we talked about these mast cells spit out all their granules and they have to remake all their granules. So they asked during re-degranulation, so a second degranulation episode, do the cells that took up neutrophils function any differently? And the answer was yes. They are better at degranulating the second time. So, sorry, Brianne, I think that your mast cells that eat the neutrophils were making it worse.
I know, I think so too. Well, I was actually making a face. Would you call this a innate mast cell memory?
Memory, huh? I did not think of that, but that is really interesting.
Is this another kind of trained immunity situation? And now we're seeing the trained immunity version of a mast cell?
Yeah, because it's better degranulation the second time.
Yeah, the second time. And it has made some kind of change after experience.
Yeah.
When it's better the second time, is the neutrophil still there or is it gone?
The neutrophil's gone by that time. So the way they did this experiment was they sensitize them, degranulate them, let them eat the neutrophils, let them rest for a few days, and then induce degranulation again. And they asked, what is the quality and quantity of the degranulated material?
I really love this experiment where they did the pull down of mast cell ribosomes versus and then looked at the transcripts that were in the ribosomes. It kind of leads to the question, well, what if something's not actively being transcribed or translated at that moment? You're obviously biasing this towards actively translated.
Yeah.
Part of me wonders about whether that matters here, but still the idea of this pull down is so cool.
Yeah, and I think people do this. I think it's a relatively routine thing, but it was used in such a creative way for this experiment because there was no other way to do this experiment. Right. Because otherwise you'd capture all of the neutrophil transcripts as well as the mast cell transcripts. And you really only want to look at the mast cell transcripts in cells that have a neutrophil or don't. And that allowed you to do that, which was really cool.
Yeah, I know. Fabulous.
Yeah. So in addition to this re-degranulation assay, they also looked at a few other things. And I will say, I don't fully understand why, but the cells had lower fatty acid accumulation, and that didn't seem to surprise them. I guess the directions that the fatty acid metabolism genes went, they expected them to have lower fatty acid accumulation. But I don't really know what the role of fatty acid accumulation is in the mast cell generally, because that was a Bodipi stain. But they do talk about getting fatty acids into the mitochondria to do the respiration stuff. And that was a bad way of saying that, sorry. But
Yeah,
they did.
I feel like I'm just wrapping my mind around some of the immunometabolism details in other cell types. And so the idea of what's going on with immunometabolism in mast cells is beyond the scope of my knowing things.
Yeah, I definitely had never thought about mast cells and how they consume oxygen and glucose use. But nonetheless, they found that the mitts had lower fatty acid accumulation, but they had higher oxygen consumption rate, especially higher maximum respiration and spare respiratory capacity. So it's kind of like what happens with macrophages when they get inflammatory. So these mitts seem to be somewhat more inflammatory. So they did some studies with carnitine palmitoyltransferase 1A that transports fatty acids to the mitochondria. And if they blocked that pathway, the mitts survived better than the mast cells. And they sort of argued that this indicated that the mitts were better able to survive in low nutrient conditions. And that, and they showed that as well. They starved them basically, and the mitts survived better. So they're arguing that probably at least one role for this is this traditional idea that they ate a neutrophil to take advantage of its nutrients and be able to survive better long term. So the last thing that's kind of neat is coming back to the neutrophil extracellular traps and things that macrophages do that mast cells don't do. And it turns out that when the mast cells take up the neutrophils and form these mitts, their increased capacity to degranulate also correlates with a higher production of pro-inflammatory lipids and release of material that's only from the neutrophil, including DNA that's typically associated with nets. So the idea here is maybe mast cells can make sort of semi-neutrophil extracellular traps because they spit out the neutrophil nets. So it's almost like they captured Spider-Man and then made Spider-Man shoot out his web. So they call this nexocytosis. So the idea, again, is that these mast cells degranulate, they cause the neutrophils to swarm, they capture one or two of those neutrophils inside, hold on to them, keep them alive for a while, and then are able to exploit their ability to make extracellular traps, because when they spit them out again, the neutrophils can spit out their traps, and they call it this nexocytosis.
So what fraction of neutrophils are taken up by mast cells? Do we have any idea?
Very, very few. Mast cell or neutrophils will outnumber mast cells hugely. And this is a rare event. I didn't say the percentages, but if I remember from that figure, it was around 6%-ish of mast cells take up a neutrophil. Okay.
So it could have an impact.
So it could, yeah.
I mean, if it were less than 1%, I would worry, right? But 6% is okay.
In the tissue, about 6% of the mast cells will contain
in a local environment, that could be a big change.
Yeah. It's not going to be 6% of your mast cells throughout your whole body have a neutrophil in them, but this would be locally when the mast cells are degranulating and the neutrophils are in the area. The last thing they did was to characterize the secretome. So when the mast cells degranulate, or the mast cells with the neutrophil in them degranulate, they identified about 25 proteins that were released by the mitts that were not released by the mast cells. So this suggests that there was something inside the mast cell that it was able to release that was unique to the mitts that wasn't present in the mast cell. So you say, well, might that be a neutrophil protein? Well, yes, 16 of the 25 proteins that they identified were actually neutrophil proteins, including like the GFP that they use to label the neutrophils. So they know it only came from the neutrophil. What this says is that there were truly proteins that were retained inside of the mast cell that when it re-degranulated, it could secrete, that were only from the neutrophil. So it's endowing the mast cell with a potential to secrete entirely new things that it could not do without having taken up a neutrophil. The other 9 proteins were histone proteins that very likely were from the neutrophil, but they couldn't absolutely say that because those proteins are also expressed in mast cells, although they should be in the mast cell nucleus, not in their granules.
Right.
So the idea here is that the mast cells can take up the neutrophils and then use what the neutrophil used in its next round of defense mechanisms that employs.
So basically they're getting both nutrients and function.
Correct. Yep. Now, the last series of experiments they did was to say, okay, great. So they can make these nets. Nets are known to induce interferons, full circle to our snippet earlier, but interferons have signaling capacities on lots of different cells, and some cells that are in the general vicinity that might respond to DNA as well as interferon that gets spit out by these mast cells are macrophages, which we come back to macrophages and innate immunity again. And what they do is they take the supernatants from degranulated mits or mast cells. So they either have a neutrophil in them or they don't. And they put them on macrophages. And they show that the macrophages now have a signature that indicates that they are responding to those components like DNA and interferon. So it's not innocuous that that the mits are secreting out the neutrophil components. They are actually having a function potentially on the cells in the microenvironment. And they mapped back the gene expression profile that they saw in these macrophages to CGAS sting pathway. I had a little trouble conceptualizing how all of that worked, but the bottom line was that the mits are secreting things that seem to trigger innate signaling within the macrophages that might involve CGAS, but there's a gap in how something outside the cell gets into the cytoplasm to signal CGAS, and that part they left out. So I'm not sure how that would happen. But overall, this is really not only cool because mast cells are eating the neutrophils. They're big cells. They are able to swim around inside the mast cells, but they're actually endowing these mast cells with new functionalities that are typically associated with neutrophils.
So they say at the end, Cindy, this interaction should be considered for the biology of long-lived MCs in the context of chronic allergic inflammation and immune disorders. So how do we do that? I don't quite know.
Well, so my thought about that is if I think about my ragweed allergies,
Right.
Yeah.
let's imagine for a second that in 2013, I had 6% of my mast cells in around my lungs, in my respiratory tract, became these new kind of mast cell. I'm going to have another year, 2014, where I'm going to respond to ragweed. And you might imagine that I'm going to pick up another set of my mast cells are going to be responding to this. And so you could imagine maybe over time, if these they don't really talk about here, how long do those mast cells stay changed? But you could imagine that those mast cells could be making up a larger and larger fraction of the mast cells, and they could really change the disease in a more chronic phase.
So if we could regulate this uptake, that could help you.
It could potentially help. It could potentially, or if we could sort of make that not happen, perhaps that could keep the allergies from maybe progressing as much.
Got it.
Is how I read that. But
Right.
I could be, I don't know if that's what they were thinking.
So mast cell related immune disorders. What's that? Is that allergies basically, or something more?
Mostly allergies. There
Asthma.
are a few other mast cell mediated diseases. Yeah, asthma for sure.
Okay.
What I started to think about is what about, let's say you get a viral respiratory infection and you have some tissue damage and some neutrophils go in, but you also have allergies and it's allergy season. How does that impact what's going on? I don't know, but it's an interesting thing to think about.
Yeah.
Or if you get pneumonia and you also have allergies, what happens there? Because those are instances in which you will have significant migration of neutrophils into the tissue to help deal with the bacterial infection,
Right.
or the tissue damage. And if you have ongoing exposure to allergens, how does that influence it? And actually, I just thought, like, what about people who just are allergic to, like, the mites and dander and things, where they might be triggered at a low level all the time?
Yeah. Yeah.
And what happens when they have a bacterial infection? I think there's a lot of interesting things that could be investigated.
I do too. And I think this also is a reminder, and I am so guilty of this. But part of that is because I teach undergraduates, that we often think about, this is the antiviral response, and these parts are responding and everybody else is not doing anything. Or we think about them and we ignore everybody else. And so the mast cells are important in allergies, and the neutrophils we ignore those. And the neutrophils are really important in phagocytosing bacteria. And we kind of don't think about the fact that this is a network that all evolved together. And so, these responses are not as separate as we often think about them. And I know I talk about them and oversimplify to kind of say this is the bacterial, this is viral, this is allergy. But here we see lots of situations where they're probably, as Cindy points out, lots of situations where they're both happening at the same time. Yeah. But even if they're not, even if you don't have a bacterial infection during your allergy response, those neutrophils, those other cells are still there. They co-evolved with all of these other responses, and they probably are doing something important.
I think you raise an important point that when we teach and learn immunology, we compartmentalize things. And it is challenging to think about how these systems interconnect.
Absolutely.
And I often ignore the fact that mast cells are actually pretty important in inflammation in general, especially when neutrophils are involved, because they do respond to pathogen-associated molecular patterns. They do degranulate and change the vasculature. They're a major contributor to vasodilation and recruitment of neutrophils into the tissue.
And they're responding to and kind of integrating with a lot of those chemokine responses that are changing recruitment of other cells.
Right. So thinking about mast cells only related to allergy, I think, is doing a disservice, because I do think that they're involved quite frequently when neutrophils are normally being recruited into a tissue.
I guess I won't always think of them as the bad guys.
Okay. Thank you, Cindy.
Yeah.
It's Immune episode 83, show notes microbe.tv/immune. And if you have any questions or comments or suggestions, we'd love to hear them. Immune@microbe.tv. If you enjoy these science programs, we'd love to have your support. Microbe.tv/contribute. Cindy Leifer is at Cornell University, at Cindy Leifer on X. Thank you, Cindy.
Thank you.
Brianne Barker's at Drew University, BioProf Barker on Blue Sky. Thank you, Brianne.
Thanks. I learned a lot. I'm going to have to rewrite some of that syllabus.
Redo your lectures, yeah.
Yeah.
It's always good. That's
absolutely.
I find podcasting really improves your courses.
I think it does as well.
And writing a textbook, I don't have to look far for new material. It's all it's really good. I'm Vincent Racaniello. You can find me at microbe.tv. Music on Immune is by Steve Neal. Thanks for listening to Immune, the podcast that's infectious. We'll be back next month.